This is Dr. Christopher Sernaque hosting episode 22 of season 6 of the Current Topics in Science podcast. This podcast will address breaking scientific news in light of the origins debate and host interviews with scientists. Today, we're going to be talking with Dr. Jonathan McLatchie about his new article, The Eukaryotic Cell Cycle, an Irreducibly Complex System. This podcast is available on the following platforms: iTunes, Audible, Google Podcasts, Spotify, and iHeartRadio. Video recordings of the podcast will be uploaded to YouTube. Enjoy the podcast. Today on Current Topics in Science, we are honored to host Dr. Jonathan McLatchie. Dr. McLatchie hosts a bachelor's degree in forensic biology from the University of Strathclyde, a master's degree in evolutionary biology from the University of Glasgow, a second master's degree in medical and molecular bioscience from Newcastle University, and a PhD in evolutionary biology also from Newcastle University. Dr. McLatchie previously served as an assistant professor of biology at Sattler College in Boston. In addition to his academic achievements, Dr. McLatchie has participated in over 40 moderated debates with representatives of atheism, Islam, and other worldviews, engaging in thoughtful dialogue on science, faith, and philosophy. He's the founder and the director of TalkAboutDoubts.com, a unique organization that provides free, private mentoring for Christians wrestling with doubts about their faith, as well as for ex-Christians exploring a rational path back to belief. You can learn more about Dr. McLatchie and his work at TalkAboutDoubts.com and JonathanMcLatchie.com. His scholarly papers are also featured on the Discovery Institute website and Evolution News. And now, without further ado, good morning, Dr. McLatchie. How is your day, and how are you doing?
Great to be here. Thanks so much for having me. Yeah, day's going well, thank you. Just got up not too long ago. It's only 8 in the morning here at the time of recording.
Well, thank you for coming on Current Topics in Science. And since this is Current Topics in Science, let's take a look at this week's current topic. You published a peer-reviewed paper in the Journal of Biocomplexity titled Phylogenetic Challenges to the Evolutionary Origin of the Eukaryotic Cell Cycle. And you wrote 2 articles for Evolution News exploring the same topic. Could you please provide our listeners with a summary of your work and explain what led you to conclude that the eukaryotic cell is best explained by the scientific theory of intelligent design?
Absolutely. So the eukaryotic cell division cycle is the process by which the cell divides. So eukaryotes, for listeners who are not biologists, are essentially cells that contain a nucleus, as opposed to prokaryotes that don't possess a nucleus. And the mechanisms of eukaryotic cell division differ drastically from the mechanisms of cell division in prokaryotes. There's almost nothing in common, either in terms of the protein components that are involved or the underlying logic. They're a complete universe apart. And it turns out that the eukaryotic cell division cycle, and indeed the prokaryotic cell division cycle for that matter, are irreducibly complex. Irreducible complexity was a term that was coined, of course, by Michael Behe in his 1996 book, Darwin's Black Box. And basically, it refers to complex systems in biology, biochemistry, that are comprised of multiple well-matched interacting components, each of which contributes to the system's overall function. And if you were to remove any one of those parts, then the system as a whole ceases to work. And so the challenge to neo-Darwinian evolution is, how would you build or assemble such a system one step at a time by a neo-Darwinian stepwise pathway without having foresight of the target? Because natural selection can't preserve some future function. It can only preserve current function, current utility. Irreducible complexity is not merely a challenge to neo-Darwinian evolution, but is also a positive argument for intelligent design as well. Because we know in every realm of experience that only conscious agents have the ability to visualize complex higher level objectives and then to bring everything together needed to realize that complex higher level objective. Whereas unguided stochastic processes are not able to do that. So I was interested in this question of irreducible complexity as applied in particular to the eukaryotic cell division cycle. And the eukaryotic cell division cycle is comprised of a number of well-matched interacting components, each of which is essential for the system to work. So, for example, the condensins are necessary to allow the chromosomes to condense in preparation for mitosis, which is a culmination of the eukaryotic cell division cycle. As the cell enters mitosis, the chromosomes become highly condensed and they take upon themselves that characteristic chromosome structure. And they're highly conserved across eukaryotes, these condensin proteins. So condensin 1 is active during late prophase and it contributes to the structural integrity of the chromosomes following the breakdown of the nuclear envelope, whereas condensin 2 functions earlier in prophase and is involved in the initial stages of chromosome condensation in the nucleus. And so these condensin molecules are composed of 5 subunits. So you've got the SMC, which stands for structural maintenance of chromosomes proteins, SMC2 and SMC4, which possess ATPase activity, and SMC proteins possess coil domains, which are essentially long, flexible arms that fold back on themselves and it creates a V-like structure. They have a hinge domain that facilitates the dimerization of the 2 SMC proteins. And they have head domains that contain ATP binding and ATPase sites, energizing the activities of the condensins. In addition to the SMC subunits, there's also 3 non-SMC subunits which bind specific regions of the DNA and assist in regulation of the activity of the condensins. So the condensin complex is basically loaded onto chromatin and in a stepwise manner, and that's directed by the non-SMC subunits. And the SMC subunits create loops in DNA, utilizing their ATPase activity. And these loops are stabilized and condensed into mitotic chromosomes. So the condensin proteins are absolutely crucial for the process of cell division. If you didn't have them, then the consequence of that would be chromosomal disorganization, as well as great difficulty in achieving proper segregation during mitosis. So that's the condensin proteins. You also have the kinetochore complex, which is a complex of proteins that assembles around the middle of each chromosome, around the centromere. And it's, again, absolutely crucial to the process of mitosis. So each of the kinetochore complexes that assembles around the centromere of each chromosome acts as an attachment site for the spindle microtubules, which radiate from structures known as centrosomes at the cell's poles. And these kinetochores assist with alignment of chromosomes at the equatorial plane of the cell during metaphase, and that ensures an equal distribution of genetic material. Kinetochores also sense tension that's generated by microtubule pooling, and thus they ensure proper attachment. And if there is improper attachment, so for example, if the kinetochores of both sister chromatids are attached to the same pole, then these errors can be corrected by the kinetochore-associated machinery. So if there were no kinetochores, if we were to knock those out, then that would result in an improper attachment of the chromosomes to the spindle apparatus, and the genetic material will be unequally distributed to the daughter cells. And they're found, these kinetochores are found ubiquitously throughout all eukaryotic organisms. There's also the anaphase promoting complex or cyclosome, which is responsible for driving entry from metaphase through to anaphase of mitosis. So mitosis is comprised of a number of different phases. You've got prophase, prometaphase, metaphase, anaphase, and telophase. And then you have cytokinesis, which is the process by which the cell actually divides. Now, at metaphase, the chromosomes are aligned along the equatorial plane of the cell. And then during anaphase, the sister chromatids, which are linked together by the centromere, are pulled apart by these microtubules. And what drives that is there's a protein called separase that is responsible for cutting the cohesin ring that tethers the sister chromatids together. Separase, its activity is inhibited by another protein called securin. And securin is the target for an enzyme called the anaphase promoting complex, or cyclosome, which acts to attach ubiquitin peptides to substrates, including securin, and that tags it for destruction by the cell's proteasome. And so when the the proteasome recognizes the ubiquitin tags, it goes and destroys securin, and now separase is liberated because securin is no longer there to inhibit it. So separase goes and cleaves the cohesin ring, tethering sister chromatids together. And so the sister chromatids then separate and they're pulled towards the poles of the cell. And whole process is actually regulated by a mitotic checkpoint known as the mitotic checkpoint complex, which is responsible for the spindle assembly checkpoint, to make sure that that only is allowed to happen once you've achieved proper association between the microtubules and the kinetochores. And so that's absolutely, these proteins are absolutely essential. So if you didn't have separase, then the sister chromatids would fail to separate, of course, and the cell would be unable to segregate its chromosomes at anaphase. And in fact, experimental knockout studies have shown that deleting separase results in embryonic lethality. So cell cycle progression would also be halted in the absence of the anaphase-promoting complex or cyclosome as well, which would inhibit the progress from metaphase to anaphase. In fact, experimental studies knocking out APC2, which is a core component of the anaphase-promoting complex in mice, for instance, resulted in lethal bone marrow failure within only 7 days. Another class of proteins that's essential for mitosis are the aurora kinases. These are essential for proper spindle formation as well as chromosome segregation. So aurora kinase A, for example, phosphorylates proteins, as kinases do. They phosphorylate proteins to drive a response, often a conformational change. So the aurora kinases phosphorylate proteins that are involved specifically in microtubule organization and facilitate the accurate attachment of microtubules to kinetochores. And in fact, mice that have their aurora A's knocked out die early during embryonic development during the 16-cell stage. And these embryos that have a defect in or missing, have a deletion of aurora A kinase, have defects in mitosis, especially in the spindle assembly. And so this indicates that the functions of aurora A are crucial in mitosis. You've also, of course, got the microtubules, which I've already alluded to. And these radiate, as I said previously, from the centrosomes and the anchor to the kinetochore complex that are assembled around the centromere of each chromosome. So during metaphase, as I said before, the chromosomes are aligned along the equatorial plane or the midpoint of the cell, and they're bound to microtubules at the kinetochores. And then at anaphase, the chromatids are pulled apart by the microtubules, driven by polar spindle forces. And so these are absolutely essential for segregating the sister chromatids into the daughter cells. So if you didn't have the microtubules, then it would severely impair the mitotic spindle assembly, and chromosome alignment and segregation would be severely inhibited. And in fact, experimental studies with the mouse embryos that are deficient in gamma tubulin exhibit a mitotic arrest that arrests development at the moral blastocyst stages. So absolutely crucial. Again, you also have the contractile ring, which is responsible for the endpoint of the cell cycle, which is cytokinesis. So this is the final stage of mitosis where the cell physically divides into two daughter cells. And it's principally composed of actin filaments and myosin 2 motor proteins together with other regulatory proteins. And these components form a dynamic bell-like structure beneath the membrane at the equator of the dividing cell, and it produces the contractile ring, generates the force that's needed for the ingression of the cleavage furrow. So myosin 2 interacts with actin filaments in the ring to generate the contractile force, and that process is energized by ATP hydrolysis. So the ring, as it tightens, it pinches inward to the plasma membrane, and this results in the cytoplasm dividing. So if you didn't have the contractile ring, then of course the cell wouldn't be able to divide, and you would have binucleated cells. So you've also, of course, got an important role of motor proteins, which are responsible for the formation of the mitotic spindle and the positioning of the chromosomes. And these are, of course, also absolutely crucial. If you didn't have those, then that would very severely compromise the transport and positioning of the chromosomes, and result in chromosomal misalignment during metaphase and difficulty in establishing the mitotic spindle. And so the result would be chromosome segregation errors during anaphase. And then, of course, you've also got CDK and cyclin molecules. So CDKs stands for cyclin-dependent kinases, and these are responsible for phosphorylating substrates to drive entry into the next phase of the cell cycle. And these are themselves activated, as their name suggests, by cyclin molecules, which bind to the CDKs. Although not all CDKs are necessary for the cell cycle to work, for cell viability, you do need at least CDK1. So and the and it is activating a cyclin molecule. And of course, you also have the checkpoints I already mentioned in passing the spindle assembly checkpoint. There's also the restriction checkpoint that determines whether the cell's ready to enter another or commit to another round of the cell cycle. There's also the DNA damage checkpoint. And if those were knocked out, then that would also cause significant problems. And so the cell cycle is clearly irreducibly complex. And so it's quite striking then that previous phylostratigraphic analysis has indicated that the vast majority of the mitotic components were already present in the last eukaryotic common ancestor. And this is interesting because, as I said before, the process of cell division in prokaryotes is very different, vastly different from that found in eukaryotes, both in terms of the protein components as well as the underlying logic. And this is surprising, I think, from an evolutionary point of view. In fact, in my paper, I did an analysis on many of the mitotic components to try to identify remote homologs of these components among prokaryotes, in particular among a class of archaea known as the Asgard archaea, which are believed to be the closest living relatives of eukaryotes. So this would include Loki archaeota, Thor archaeota, Odin archaeota, and Heimdall archaeota, named after Norse deities. And basically, my paper shows that for the vast majority of the mitotic components, you cannot find remote homologs of these, even among the closest living relatives, or at least thought to be the closest living relatives of eukaryotes among prokaryotes, among the archaea. And so how would you go from, and so the origins of the eukaryotic cell division machinery seems to be very closely associated with the origins of the eukaryotic cell itself. The components seem to have arisen after the split between eukarya and archaea. Between eukarya and prokarya. And so how would you go from the prokaryotic cell division machinery, which is itself irreducibly complex, to the eukaryotic cell division machinery, which is itself irreducibly complex, without passing through any maladaptive intermediates, particularly given the very narrow, or relatively narrow time window that you have, and generating basically the vast majority of the components de novo. So this is why I think that the eukaryotic cell division cycle poses a significant challenge to the evolutionary process, but makes far better sense from a design perspective.
Thank you for that detailed explanation. It reminds me of when you said in your article, The Eukaryotic Cell Cycle, the Irreducibly Complex System, where you concluded by stating, any system that achieves a complex, higher level objective by means of various well-matched interacting components requires foresight to come about. So as somebody who studied biology through a forensic lens, you have unique insights into the nature of foresight. Could you elaborate on the connection between foresight and the development of intricate systems like the eukaryotic cell?
Absolutely. So foresight is something uniquely associated with intelligent agents such as ourselves, where we can visualize complex higher level objective. So, and there's many systems in biology that seem to require a cause with foresight to bring about. For example, one thing that I've written on recently is the process of apoptosis, by which our digits form during embryogenesis, right? During embryogenesis, our digits form through a process of programmed cell death. The technical term is apoptosis. And the question is, from a neo-Darwinian point of view, how would you select for a process involving programmed cell death without knowledge of where the target lies, right? And so it points to a process of design, and so it points to a process that has foresight, that can visualize a complex end goal, and that's really the case for all irreducibly complex systems. They require a process with foresight. So another example would be the DNA replication machinery, which is part of the cell cycle, right? So it's the cell cycle is you've got most of the cell cycle is taken up with what's collectively known as interphase, where you have G1, S, and G2. So G1 and G2 are the G stands for gap, so it's gap phase 1 and gap phase 2, and between those you have what's called S phase, which stands for synthesis, which is where the DNA is being replicated or synthesized in preparation for cell division. And the DNA replication machinery itself is irreducibly complex. So you have the DNA polymerase, for example, that actually performs the copying, but you also have to have the DNA helicase that breaks the hydrogen bonds on the DNA double helix to prepare for copying, and you have to have single-stranded binding proteins that bind to the single strands of DNA to prevent them from reannealing during copying. You also have to have the sliding clamp that clamps the polymerase onto the strand of DNA to prevent it from falling off during copying. You have to have the clamp loader that loads on the sliding clamp. You have to have the topoisomerase enzymes, because as a result of the torsional stress on the DNA molecule, the DNA forms supercoils. You think about an old telephone cord that forms supercoils, and if this is left unchecked, it can result in the DNA breaking and DNA replication would stall. And so the topoisomerase enzymes is a class of proteins that basically are capable of alleviating these supercoils by cutting one strand, passing the other strand through the gap, and then resealing the break. You have to have the primase enzymes, because the DNA polymerase isn't able to begin copying without the presence of an available 3' hydroxyl group. And so the primase, which is a form of RNA polymerase, synthesizes a short sequence of RNA from which the DNA polymerase can then extend. Because of the antiparallel nature of DNA, with the two strands of DNA running in opposite directions in terms of their polarity, one strand, namely the lagging strand, has to be replicated backwards and discontinuously. And so it's literally looped around, and you synthesize short primers with a primase enzyme, and then from those you extend them to form short fragments of DNA known as Okazaki fragments, which have to get stitched together by an enzyme called ligase, and the primers have to get removed by an enzyme called exonuclease enzymes, and then replaced by DNA with DNA polymerase 1, and so forth. If any of those components were missing, and I haven't even been exhaustive in my treatment, but if any of these components were missing, then you don't have DNA replication machinery that works half as well as it used to, or a quarter as well as it used to, but it's broken. And so how would you produce that without knowing where the target is? And this is an example of what I call irreducible complexity on steroids, because DNA replication is itself necessary for differential survival, right? You cannot have natural selection unless you have differential survival. And so you can't really invoke natural selection to explain the origins of self-replicatability without assuming the existence of the very thing you're trying to account for. And so this is, I think, problematic. And I mentioned that the bacterial cell division apparatus is also irreducibly complex. Just give one example relating to that. So as the bacterial cell prepares for division, it elongates, in the case of a rod-shaped bacterial cell like E. coli or Salmonella, about double its original length. And then you have the formation of the Fitz-Z ring around the division septum, and the cell differentiates into two daughter cells. Now, in preparation for cell division, though, as I said, the cell elongates, and that elongation process involves severing the peptidoglycan cell wall. And of course, that can result in the cell bursting, or the technical term is lysing, as a result of the osmotic pressure. And so you have to actively rebuild the cell wall in a coordinated way. So there's a class of proteins called autolysins that are responsible for breaking the cell wall, severing the cell wall. And then you have to, in a coordinated fashion, rebuild the cell wall. So there's a protein, for example, called bactoprenol that shuttles the peptidoglycan precursors across the cell membrane. And then there's a class of proteins called penicillin binding proteins, which are responsible for performing the cross-linking reaction, or the transpeptidation reaction that results in the reparation and rebuilding of the cell wall. Now, as the name suggests, the penicillin binding proteins are the target for penicillin antibiotics, because they disrupt that cross-linking process, and that results in the cell bursting. And so we know how crucial these penicillin binding proteins are. And so from a neo-Darwinian evolutionary perspective, how would you select for the process to break or sever the peptidoglycan cell wall without having a mechanism in hand for rebuilding the cell wall? Because without that, the cell bursts as a result of the osmotic pressure and dies. So, I mean, there's countless examples that one could give. But irreducibly complex systems, as I said, are not merely a challenge to Darwinism. They're also a positive argument for design, because intelligent agents uniquely have that capability of foresight.
Yeah, that is a very fascinating perspective, Dr. McLatchie. So I'm hearing you suggest that unguided natural processes lack the foresight required to produce the eukaryotic cell's complexity. This reminds me of when you wrote in the abstract of your paper, while the time window available for the origin of these mechanisms is unclear, even a large window, 2 to 3 billion years of time, would be insufficient for blind evolutionary mechanisms to produce such complex systems. This analysis raises significant questions about the feasibility of evolutionary processes to account for such an abrupt transition. So, Have any of the evolutionary colleagues that you have responded to this challenge, and do you find their rebuttals to be convincing? Why or why not?
No, actually, I haven't seen any attempted rebuttals to the argument that the eukaryotic cell division cycle is irreducibly complex, or indeed the fact that the vast majority of the components of the eukaryotic cell division machinery appear to be eukaryotic de novo innovations that arose after the split with prokaryotes. I mean, I think it's a significant challenge to evolution, and I haven't seen any attempted detailed account of how you could go from the prokaryotic cell division machinery to the eukaryotic cell division machinery without passing through any maladaptive intermediate stages, and particularly without leaving any trace of transitional forms, if you will, in modern organisms. We don't see any examples of a cell that's doing partially prokaryotic cell division machinery and partially eukaryotic cell division machinery. But I mean, the two systems are just, there's a wide gulf between them, and both are irreducibly complex. And so, how would you go from one to the other without passing through any maladaptive intermediate stages, and particularly having to generate? I mean, there's no homologs for most of the eukaryotic cell division machinery proteins among prokaryotes. So you have to basically generate all of them de novo. You can't borrow from other systems because they're not functioning in other systems in prokaryotes. So it's highly problematic, I think, if you're a neo-Darwinian evolutionary theorist.
Sometimes when I've dialogued with evolutionists, they'll say, well, look at the mitochondria, right? This is an example of you have a prokaryotic organism, right? And now it's transitioning sort of, through the endosymbiotic theory, right? Now that mitochondria is now building on to the eukaryotic cell, and that's how we got something like a eukaryotic cell. What do you think about that?
Yeah, I don't find the endosymbiotic thesis particularly compelling. That idea was originated by the biologist Lynn Margulis. Basically, it's the idea that proto-eukaryotic cell or an archaeal cell engulfed a bacterial cell, and that gave rise to the mitochondria. Mitochondria is plural of mitochondrion, and that energized the transition from the prokaryotic cell to the eukaryotic cell. Chloroplasts are also thought to have arisen in a similar way through engulfing bacterial cells that formed a symbiotic relationship and ultimately became dependent upon their host cell and gave rise to these membrane enclosed organelles that we call mitochondria and chloroplasts. As I said, I don't find that thesis to be particularly compelling. There has to be a genetic basis for inheritance of the mitochondrial genes. Most of the mitochondrial genes have actually migrated to the nucleus, and it's interesting that the mitochondria actually has a slightly different genetic code from the conventional genetic code as well. So UGA, which is conventionally a stop codon, codes for tryptophan in mitochondria, for instance. And so you would have to kind of do a recoding on the way over to the nucleus. And I don't see any plausible mechanism of transferring these genes from the mitochondria to the nucleus while recoding them in the process. So I'm not particularly compelled by the endosymbiotic theory.
Well, thank you for addressing both those questions. Speaking of the cell, in your article on the irreducible complexity of the eukaryotic cell, you argued that several of the cell components, everything from the anaphase promoting complex to the CDC and cyclin molecules and the cell cycle checkpoints, are all examples of irreducible complexity. Do you have any testable predictions from the intelligent design framework about any of the components you've mentioned, or any other aspects of the cell? What avenues for future research do you see this opening up?
Well, intelligent design predicts that we will find systems in the cell that are rich in specified complexity, informational content, irreducible complexity, because these are all processes, these are all phenomena that require foresight to bring about. And of course, intelligent design is basically a synonym for foresight. And so that's what intelligent design predicts. And it's pretty much what we're finding, right, as we study the cell, whereas neo-Darwinian evolution would predict the opposite, would predict that we would not find irreducibly complex systems and systems that seem to require foresight to bring about. So I'd say that this is a confirmation of predictions of intelligent design. I think that the more that we understand, the more that we understand about the complexity of the cell, the more of this irreducible complexity we find. And so more knockout studies genetic knockout studies where proteins are knocked out, I think will reveal even stronger that these systems, including the cell cycle, are in fact irreducibly complex.
Dr. McLatchie, we have now come to our final question. It's clear that your scientific work reflects a deep commitment to understanding the intricacies of life and the universe. Now, while intelligent design is a scientific theory, it's not a religious doctrine, you are also a person of faith, a Christian. How does your faith shape your journey as a scientist, not just in your work, but in the way you see the beauty, the complexity, and the purpose in the world around you?
The psalmist says that the heavens declare the glories of God, the skies proclaim the work of his hands. Paul, the apostle, in Romans 1, says that God's nature, his eternal qualities, divine nature being clearly seen, being understood through things that have been made, so that men are anapologetes, without an apologetic, without an excuse. And I think that Paul was right. I think that the evidence in nature, both in the physical and life sciences, points forcefully towards the reality of cosmic creator. And in fact, throughout most of human history, the vast majority of people have recognized this. And it's been widely taken for granted that God exists. In fact, many of the critics of Christianity back in the day, in say the 1700s, were deists rather than atheists. It was widely taken for granted that, of course, there's a creator behind nature and behind the universe. And then, of course, the tide began to change, in particular with the publication of The Origin of Species by Charles Darwin, which purported to provide a mechanism by which you could account for the appearance of design without recourse to an actual designer. And if Darwin got it wrong, as I contend that he did, then we're back to where we were before Darwin, where the design is, in fact, apparent in nature. God, in the broad sense, is not hidden, as is sometimes contended, is revealed through the created world. And if Darwin got it wrong, then, as I said, we're back to where we are before Darwin. And the challenge to naturalism in the case for design in nature has been has become far greater since the time of Darwin. In view of our modern advances in the life and physical sciences, we understand the genetic basis of life, the fact that what runs the show in biology is digitally encoded information content. We have a language convention in the form of the genetic code and so forth. And we have irreducibly complex molecular machines that govern the workings of the cell, etc. I think that that provides an answer to the problem of divine hiddenness, at least in the broad sense, that no, God is not hidden. And yes, Darwin gave a technical response to that design intuition, and so it has to be met with a technical rejoinder. But that doesn't mean that the inference itself is technical. I think that it can be grasped tacitly or implicitly. And I think most Christians or most theists do grasp that inference tacitly or implicitly. And so since there's, I think, an abundance of evidence in the natural world for theism, I think it makes it incumbent upon us to investigate the public evidences of Christianity to discover whether God has in fact revealed himself in the person of Jesus Christ. And I also have strong expertise in New Testament studies in particular. And so I'm particularly compelled by by five major lines of argument for the truth of Christianity. These would be the case for Jesus' resurrection, the conversion of Saul of Tarsus, the Christological trilemma argument, the argument for messianic prophecy, and the argument for modern miracles. Each of these five arguments I find to be compelling in their own right. When you take them in aggregate, I find it to be essentially decisive. And so, yeah, that is essentially why I'm a Christian.
Dr. McLatchie, thank you very much for your time. It was an absolute joy. And to our listeners, thank you for taking the time to learn with us on current topics in science, where scientific discoveries are examined in light of the origins issue. You can find Dr. McLatchie's biographical information, his lectures, papers, and more information at talkaboutdoubts.com and jonathanmcclatchie.com, which are both linked in the description, along with a link to the Christ Jesus Ministries merch store. Please share and subscribe to the Current Topics in Science podcast. It's available on iTunes, Audible, Google Podcasts, Spotify, and iHeartRadio. Thanks again for listening, and remember, the truth saves.